Hi. Hello everybody. My name is Iwijn De Vlaminck. I'm a postdoc in the lab of Steve Quake at Stanford. And today I'm going to tell you a little bit about how we've been using DNA sequencing for the sequencing of plasma DNA, and that is cell-free DNA or circulating DNA, for the diagnosis of, simultaneous diagnosis of rejection and infection in organ transplantation. And this work came out of a very strong collaboration, I think we can say, between our lab and several people at Stanford Hospital that have an interest in heart and lung transplantation. But before I go into describing the assay and talk about the results, I wanted to share some numbers with you about organ transplants. So there's about 180,000 people living with solid organ transplants in the US alone. There's about 2,000 heart transplants performed every year, and there's about 1,000 lung transplants performed every year. At Stanford Hospital alone, Stanford is a bit of a center of expertise when it comes to transplantation. At Stanford Hospital, there's about 60 heart transplants performed every year, and about 40 lung transplants are carried out. Now, all these patients require post-transplant care, and post-transplant care in the context of transplantation is challenging, and it's challenging because these patients face a dual risk of rejection and infection. It's only since the early 80s, with the advent of very strong immunosuppressants, that transplantation has become a viable therapeutic option. So via immunosuppression, you can mitigate pretty much the risk of transplant rejection. But immunosuppression or loss of immune strength then opens up the window for a new set of problems in the face of infections. And so really the therapeutic window that is available for treatment of these patients is very, very narrow. You can imagine that in this context, it's very important to have very informative diagnostics at hand, but it turns out that none of the metrics that I've put on the axis here can be measured faithfully. It's fair to say that right now there's really no way to measure somebody's immunocompetence or the level of a transplant patient, for example, or any patient for that matter, level of immune suppression. It's of course true that there's a lot of informative measurements available for infection, for example, molecular assays based on PCR, but most of these assays require on a particular hypothesis for what the source of the infection might be. For measurements of transplant rejection, also there's a lack of informative diagnostics. I'll just show you the example of the heart transplants. In heart transplants, the current gold standard for rejection diagnosis is the endomyocardial biopsy. And the endomyocardial biopsy is a surgical procedure in which pieces of tissue are removed from the transplant graft and then stained and examined under a microscope. So you can imagine that this is a very subjective measurement. It turns out if you ask two pathologists to grade the same biopsy slide, that they will only agree in about 70% of the cases. It's also an expensive procedure. It's invasive with major complications in about 1% of the cases. But nevertheless, for example, at Stanford in the first year post-transplant, heart transplant patients will on average undergo 10 of these biopsies in the first year. And so there's clearly a lot of room for improvement, and an opportunity for such improvement is found in this phenomenon of circulating cell-free DNA. Cell-free DNA is DNA that is not contained within cells and is floating around in blood. It's usually associated with histone octamers. And it is prevented from degradation by nucleases that are abundantly present in blood because of its tight association with with proteins or histone octamers. It was already discovered in 1948, even before the structure of DNA was resolved, and that's because it's abundantly present in plasma. There's about 10,000 genome copies worth of cell-free DNA in every milliliter of blood. So that means there's about 100 billion fragments of DNA available for analysis via sequencing, for example. The mean half-life time of cell-free DNA is short, 15 minutes. So that means that the turnover rate of cells must be pretty high in order to sustain this steady state level of cell-free DNA in blood. Already 15 years ago, Dennis Lowe found that in the context of organ transplantation that there's actually donor-derived DNA molecules circulating in plasma. And he made that observation based on sex mismatched patients, in particular female recipients of a male graft, where he showed that there's actually chromosome Y derived DNA floating around in blood. And already in his seminal paper, he proposed that you could use donor DNA as a marker of rejection. And the idea is simple here. The idea is that when rejection occurs, there will be damage to the graft, and as a consequence, cells within the graft will die and their genomes will end up in the bloodstream. And so you can imagine that if you can count the number of donor molecules versus the number of recipient molecules, that you can come up with some kind of metric of graft damage. And our lab, just a couple of years ago, proposed this new technique that is based on genotyping that would allow to measure the fraction of donor DNA and the presence of donor DNA in the blood of transplant recipients, regardless of the fact of whether the transplant recipient is sex matched or sex mismatched with the donor. And so the idea is very simple. We collect genotypes from both the donor and the recipient pre-transplant, and then we use sequencing of cell-free DNA to establish the number of donor-derived DNA molecules post-transplant. And so you can imagine that if the donor and the recipient are homozygous at a particular position, so let's look at, say, position N in the diagram here, both the recipient and the donor are homozygous and carry the C allele. Are homozygous but carry a different allele. The donor carries the C allele and the recipient carries the A allele. So you can imagine that if you sequence all the plasma DNA and you find a molecule, a DNA molecule, which sequence maps to position N in the genome, and you find an allele C, that you can have some confidence, or that you can evaluate with some confidence that that molecule must come from the donor's genome. And we'll talk a little bit about error rates in just a second. So this is essentially the basis of the measurement. So what we've done over the last couple of years is we've collected a lot of samples from both heart and lung transplants. And I'm first going to talk about the heart transplants. In heart transplants, we've recruited about 20 pediatric patients and about 45 adult heart transplant patients. And we've collected whole blood samples pre-transplant for genotyping, and then plasma DNA or plasma samples post-transplant. And we've purified the plasma DNA and sequenced it. And eventually that allowed us to evaluate the fraction of donor-derived DNA molecule, which is the metric that we are interested in, and we compared that to the biopsy grade. We've collected about 20 million sequences per sample. That means that we've sequenced about 8 samples per high-seq lane, and there's about 50,000 informative SNPs available in the genome. That means that 50,000 SNPs for which the donor and the recipient are homozygous and carry a different allele, so it makes it very straightforward to distinguish both donor and recipient-derived molecules or sequences. And about 15,000 of our sequences, we do shotgun sequencing again of the plasma DNA, align randomly to these informative SNPs, and allow us to count donor versus recipient molecules. One important aspect of the assay is that we're able to measure the error rate for every individual sample. And the way we do that is by evaluating matched SNP pairs. And that is, we look for SNPs for which the donor and the recipient are homozygous and carry the same allele. So now you can imagine that if both the donor and the recipient carry allele A at a particular position, and you measure by sequencing a C, a G, or a T at that particular strand and position, that you must have made some kind of error, right? And so it's possible to evaluate the error rate, and that error rate measurement was important, it turned out, to not only exclude samples that had suffered from high error rate, but also it allowed us to figure out how the measurement errors coupled into our measurement signal, and allowed us to do some background correction on the basis of that observation. And so all of the measurements I'll show today are essentially measurements that have been corrected for that measurement error background. And so the measurement error is actually pretty low. We get it wrong in about 4 out of 10,000 cases, or 4 out of 10,000 measurements. But let's go to the actual measurements. I think it's most interesting. These are some first data for patients that do not reject. So these are all transplant patients, about 11, I think, that went on to have a rejection-free course in their first year. And so what we find is immediately post-transplant, we find an elevated level of donor DNA up to, on average, it's about 4%, I believe, of donor DNA on the first day post-transplant. And this is likely due to some damage related to surgery or some loosely associated cells, donor cells that are releasing the blood. But then very quickly, the signal drops to a very low background level. And essentially, for recipients that do not reject, we do not measure any donor DNA. However, this is quite different for transplant patients that do reject. For example, this one that goes on to have a severe rejection at month 15. You can see that there's an elevation in donor DNA around that time point. And there's some other examples. For example, this pediatric patient had multiple rejection events. And here we have a transplant patient that had rejected and required a repeat transplant as a way to intervene. And what you can see is that the donor fraction decays back to a very low baseline level after this repeat transplant. And so that indicated to us that there's very little residual chimerism, and that the signal is not confused by residual chimerism, so that you can use this test also in the context of re-transplant. We've collected many samples, and there were several hundreds of biopsies, matched biopsies available for this measurement, and we of course did a signal analysis, and we find that the donor fraction is indeed elevated at time of rejection compared to samples that are recorded at quiescence. You'll see that there's only very few outliers here, and most of the samples here recorded at quiescence for patients that do not reject actually show very low donor fraction compared to samples collected at rejection, where we see this highly elevated donor signal. You can do a receiver operating characteristic analysis, so plotting the true positive rate versus the false positive rate as you change the threshold for detection. And what you really care about is essentially the area under the curve here. The greater the area under the curve, the better the test. And what we find is that the area in the curve here is about 0.83, which is much higher than the area under the curve for another non-invasive diagnostic that is based on a measurement of gene expression markers in blood, and an analysis of the signal against the biopsy indicated that a lot of this the sensitivity of our test probably approaches the sensitivity and the selectivity of the gold standard. We looked at early detection, the potential for early detection, and we find that donor DNA signals are elevated up to 4 or 5 months before the actual rejection event. We find that the detection quality is higher for patients that are younger and for samples that are collected at a later time point post-transplant. So then in the second project, we wanted to figure out whether this test would also be applicable to other settings, such as, for example, lung transplant. So we wanted to address the question, is this test universally applicable? Is it also applicable to other organ types and other transplant settings? And so we looked at lung transplants, and the survival rates for lung transplantation are actually quite a bit lower than the survival rates for heart transplants. The median half-life time is only 5 years compared to 10 years in the case of heart transplants. And that is due to several major complications that arise following lung transplantation. For example, a syndrome called bronchial obliterance syndrome, which is related to the progressive loss of lung function, and is likely due to chronic rejection, the constant occurrence of minor rejection events. Then acute rejection, and of course infections that plague the lung, because the lung is of course exposed to the outside environment. So again, we did something very similar than we did for the heart transplants. We collected, in this case, samples mostly from adult lung transplant recipients. 51 of these 44 of these had a bilateral lung transplant. That means that they received both sides of the lung, and 7 of these only received a single lung transplant. We collected samples pre-transplant for genotyping, and post-transplant we collected blood samples and extracted plasma and plasma DNA for sequencing. And these are some of the results. And first, we again look at patients that do not reject. We find for lung transplants very highly elevated levels of donor DNA immediately post-transplant. Up to 50% of the DNA floating around in plasma of these transplant recipients on the first day is actually donor-derived, and that's much higher than what we saw for heart transplants. That signal then quickly decays to sort of an intermediate level, and then there's the signal stays elevated for several months and only decays to a low baseline after about 100 days. So there's this two kinetic decay process going on here for lung transplants. And in this insets, we're comparing the dynamics for the lung versus the heart. And you can see that there's much more donor-derived DNA for lungs than there is for hearts. It's kind of curious. It's interesting to look at the signal for single and bilateral lung transplants. And as you would expect, we find higher levels of donor DNA for transplant recipients that received both sides of the lung versus only a single side of the lung. So there's a nice correlation between transplant mass, graft mass, and the level of donor DNA in absence of rejection. And you can then extract the number of the turnover rate of graft cells per second. And it's actually interesting to see that there's about 100 lung cells that are turning over every second. That level of turnover is required to explain the levels of donor DNA that we're measuring in the study. Of course, we also looked at samples from patients that ended up rejecting. And we find that there's, again, an elevated level of donor DNA at the time of rejection, coinciding with this rejection as observed by the transbronchial biopsy. And the plot on the right shows, this one here, shows essentially all the data we've collected for patients that show signs of clinical rejection and no signs of clinical rejection. And you can already tell that there's an elevation for the data that is colored red here. And here we've excluded samples collected in the first 60 days post-transplant because we find, even for patients that do not reject, that there's an elevated level of donor DNA early on post-transplant, possibly related to decay of the lung, that parts of the lung that are not connected to the blood circulation. Okay, so we exclude those samples that's collected in the first 2 months and then do a signal analysis. And this is what comes out of it. Again, we find that donor DNA is significantly elevated at time of rejection. For patients that are moderate, have suffered from a moderate to severe rejection, we find elevated levels of donor DNA compared to patients that do not show signs of rejection, at least based on this transbronchial biopsy. And you can again compute an ROC here, and we find that the area under the curve for discriminating severe rejections from no rejections is about 0.9. And we went on to compare to various other metrics and find that there's a good correlation between graft injury as noted via various kinds of measurements in lung transplantation and donor DNA. And so the conclusions here for now are that cell-free DNA enables a diagnosis of rejection in transplantation, and that cell-free DNA potentially offers a universal way to diagnose transplant rejection across all patient groups and across all organ types. And we find that, at least for heart transplants, that the cell-free DNA test outperforms other existing commercially available non-invasive tests. All right. So, but there are some issues that are remaining. And first of all, donor DNA is not informative of over immune suppression. So there's still this need for more informative measurements of the level of immune suppression. The other shortcoming of the test is that donor DNA is not informative of infection. So it doesn't discriminate between infection and rejection related tissue damage. And there's a few examples for why this can matter. For example, in kidney transplants, there's often a lot of damage to the kidney due to polyoma virus that is actively replicating in the kidney. And similar things have been noted for infections with cytomegalovirus in the context of lung transplantation. And so, I wanted to remind you of this dual risk that exists in transplantation. And so we have a marker that's potentially informative of rejection, but it's still clear that more work needs to be done on the infection front. And so one way to illustrate this is we've collected clinical data, essentially all the clinical data available for these patients on infections. And we found that there's an elevated level of donor DNA at the time that CMV infections are diagnosed or detected in these patients, both in bronchial lavage samples and in serum. So when patients are positive for CMV, a particular kind of herpes virus that can cause graft damage, we find elevated levels of donor DNA. So this is a clear sign that there is infection-related tissue damage taking place. It turns out that CMV is the only organism for which we find evidence of robust evidence for this infection-related tissue damage. So this is something that seems to be unique to CMV and is in line with what people have observed in the transplant literature. So, but then, of course, if there can be infection-related tissue damage, the question is, can you also diagnose infections based on sequence data? And so what we did is we looked for CMV, cytomegalovirus, related sequences in our data sets, plasma DNA data sets. And we found indeed that at time of CMV diagnosis that you can find bits and pieces of the genome of this particular herpes virus. And again, we do a signal analysis and we find that there's a good correlation between that measurement based on sequencing and the measurements based that are performed in the hospital. So this is just a demonstration of the use of shotgun sequencing for the diagnosis of a particular kind of infection. We looked a little bit more broadly in our data sets we looked a little bit more deeper at the non-human derived sequences in our data sets, and we find that about 75% of the sequences that are not human derived are actually coming out of the genomes of viruses, and this is corrected for genome abundance. So this is here we're taking into account the differences in genome sizes of viruses and bacteria. And so 75% most abundant viruses are most abundantly represented in these data sets. Given that these are shotgun sequencing data, you can drill down on this at any level of classification, and so you can explore what kind of viruses are found. And we find evidence for many different viral families in our data sets. And one viral family that is abundantly present is the family of the antiviruses here, indicated in orange. So this is the orange bit of the pie here. Now it turns out that antiviruses are commensal viruses that are ubiquitous in the human population, and essentially anybody in the Western population has been infected with this virus, and we find lots of it in the plasma of these organ transplant recipients. We also find evidence of herpes viruses, for example CMV. The example of CMV was already touched upon, but also polyoma viruses, pox viruses, adenoviruses, etc., and also evidence of phages was found. You can, given as the shotgun sequencing data, you can drill down on this a little bit more and find down to the strain level. You can figure out what the most likely cause of these DNA of these reads or sequences are. And you can do something very similar for bacteria. Look at different levels of classification. You can look at the different bacterial phyla and bacteria orders that are represented in the data sets. The most interesting finding, observation from this study was that that the virome, so the collection of different viruses that are found in the plasma of these transplant recipients, that the structure of that virome, the composition of that virome, is very strongly affected by the immunosuppressant and antiviral drugs that the patients receive. So when patients are very strongly immune suppressed, and this is typically after 3 or 6 months post-transplant, we find a lot of these antiviruses. So this is the orange family here, indicated in orange, in plasma. And so it seems that antiviruses are particularly good at taking advantage of loss of immune function. And this is in line with what some other folks have observed, for example, with HIV patients that progress towards AIDS. It's found that there's more and more antiviruses present in plasma. You can look at the time dynamics of the virome structure, and immediately post-transplant, we find only a small fraction of antiviruses. So these are the orange guys here. They're only represented as a minority fraction. But after 3 months or so post-transplant, you can see that the antiviruses have completely taken over and are now the most abundantly represented species. After 6 months post-transplant, so this is when the drug doses are at the highest level. This is when the risk of rejection is highest. So patients are most deeply immune suppressed. After 6 months, it's judged that the risk of rejection is a little lower. So in typical transplant protocols, the drug doses will be tapered off, and what you see is an immediate response of the virome to that tapering of the drug doses. And so the last question we addressed was whether there's a relationship between whether the virome structure is different for rejecting versus non-rejecting individuals. And this was based on the hypothesis that rejecting individuals might have an immune system that is a little bit more active, is a little more strongly functioning. And so that would mean that you would see a lower load or lower probability of infection and potentially a lower load of various viruses. Whereas patients that do not reject may have an immune system that is more effectively suppressed, and therefore the load of viruses might be higher. So that was a simple idea we tested here. And we particularly looked at the analoviruses because we find that there's such a tight correlation between the load of analoviruses and the level of immunosuppression. And what we find is that indeed the load of an allovirus allows stratifying the rejecting versus non-rejecting individuals. So non-rejecting individuals tended to have a higher load of analoviruses, which is in line with this hypothesis that non-rejecting individuals have an immune system that is more effectively suppressed on average. And so we also then proposed that you could use the load of an allovirus as a marker of immune suppression, and we find that again that the load of an allovirus loads allow stratifying patients that that do not reject versus severe rejecting patients, and here we find an area on the curve of about 0.7. And so this brings me to the overall conclusions. We find that donor-derived DNA is an informative marker of graft injury in both heart and lung transplantation. There's some hints, and we're exploring this much deeper now, that sequencing can be used as a tool for the broad hypothesis-free diagnosis of infections. It's interesting to look at transplant patients in this context because there's a lot of clinical data on infections available to which you can compare the data to. We find that pharmacological treatment, in particular immune suppression, has a very strong effect on the structure of the virome, so the collection of different viruses that you find in plasma, the composition of the virome. And last but not least, we find that there's a strong interaction between immune strength and the virome structure, and that the load of analoviruses could be used as a potential marker for immune competence. This brings me to the acknowledgements. I particularly want to thank all my colleagues at the Quake Lab, and particularly Steve Quake for being such a fantastic advisor, and all the folks at Stanford Hospital that collaborated with us on this project. This is really a large diversity of people, in particular Karen Kush, that really was a strong lead of this project at the hospital end. And then lastly, I want to say that at early next year, I'll be moving to Cornell to set up my own lab. And so in case there's somebody in the audience that is looking for postdoc positions or other kinds of positions and is interested in this type of work, or in work related to single cell genomics, which is another thing that I will be focusing on, then feel free to contact me. And so with this, I'm open for questions. So the first question is from Rogan Rattray from Oregon. And Rogan asks, how large of a panel of genes do you analyze? So it's important to point out that for the rejection diagnosis, that we use shotgun sequencing. So we're not doing targeted sequencing of a particular gene. And we're genotyping the patient across the entire genome, and we use about 1 million to 2.5 million markers that are randomly, spread out across the genome. So we're not focusing on particular genes here. We use shotgun sequencing, random selection of sequences in the genome. I hope that answers that question. Are there any other questions? So Rebecca Fine asks, how will your data analysis impact future transplant research and patients? Well, we hope that our study and other related studies will allow to improve post-transplant care. First of all, there's a need for better and more informative markers of rejection. We hope that the number of biopsies that patients have to undergo post-transplant will be able to be reduced based on our findings. There's good hope that the cell-free DNA test that we and others have described in this format or another format will be available in the clinic within the next few years. And then outside of that, we hope that the study of the infections will allow to, for example, tailor the immune suppressant dosage to the needs of the particular patient. So, Abbas Diba asks, in analyzing of genes, do you focus on the importance of epigenetics? We haven't done so. It would be interesting to do that, but we haven't done so. So right now, we haven't looked at any epigenetic changes or markers. Nikolai Razek of University of Alberta asks, if I follow correctly, can Virome be indicative of immunosuppressive impact of the chosen drugs? Indeed, yes. So we believe that the structure of the virome, the abundance of particular virotypes can be used, is informative of the level of immune suppression of particular patients. So it's important again to point out that in transplantation, every transplant recipient receives essentially the same drug regimen, but it's clear that not everybody responds in the same way to the same drug dosage, and that leads to over immune suppression or under immune suppression for many of the transplant patients. And the hope is that by measurements of, for example, the structure of the virome or other measurements that we're working on, that it will be possible to tailor the drug therapy to the needs of the patient. So tweak the immunosuppressive dosage based on the observed level of immune competence of the transplant individual. All right. So any more questions? If not, I would like to thank the audience and wish you all a good day. Bye.